Good day. How are you? So today we are going to study the introduction of microbiology. What is microbiology and what comes under the microbiology, or what organisms does the microbiology deals with? Let's start with the definitions. Now there is a simple definition that the world knows microbiology from. That is the study of living organisms. But what living organisms? These are the living organisms that comes under the category of the microorganisms. So let's see what is the micro mean. Let me give you some fun facts. Our eyes can only distinguish two objects if they are placed 2 millimeters 1 millimeter apart. If you go below that, our eyes cannot see it. So just think about the microorganisms. Why aren't we able to see it with our naked eyes? It's because they can't be seen. Our eyes just don't have that technology in it, that machinery in it, to be able to differentiate them as different microorganisms, because they are so small that they look as one. So if we see the cluster of the microorganisms, we just see as a spot. If they are actually in millions or trillions of numbers, only then can we see them as a spot, a big spot of, just like a drop of blood or water. But our eyes cannot see deeper than that actually these are the microorganisms and they can move too. So that's why we need a microscope. Microscope actually has a technology that can see through the 1 millimeter limit. That's why it can differentiate the microorganism as the microorganism. Unlike us, that we see as non-living object, they can actually capture their movement as well. I hope you learn why the microorganisms are the organisms that need microscope to be seen. Now here comes the complex definition. It actually isn't complex in the concept, it's just difficult in the terminologies and the depth. So it is the branch of biology. Biology means it's the branch of living organisms, but that focuses only on the microorganisms, their structure, their function, their genetics, and their interaction. Now, what are the microorganisms? The microorganisms are bacteria, viruses, and protozoa and fungi. So let's dive deep and know what these microorganisms are. The first one is bacteria, and the bacteria are single cell. They are organisms that only needs one cell to become an individual that can work alone, and they are prokaryote. Pro means fake nucleus or not true nucleus. The genetic material, which is actually the chromatin material, is actually suspended in the center of the cells, mostly in the center of the cells, bacterial cells, and it doesn't have coating. And the bacteria now can be beneficial and can be harmful. We all know there can be a bad side and a good side to everything in this world, just as the light and shadow. Now, example is Escherichia coli, that is E. coli, the short term. If you are going to be a scientist, be a one that is actually one. Means they have to know the scientific. Actually, I'm asking too much from you. The learning the scientific name is actually pretty difficult. Even I don't know. I just have to remember the main ones and then go to the Google for the complex ones. So actually, Escherichia coli is a pretty common bacteria, so make sure to remember its name. These are commonly found in the intestine. Whose intestine are we talking about? It's the intestine of humans. And they don't harm us. Actually, they are good bacteria. They produce vitamin K for our body, which our body cannot produce on its own. You have to remember that everything you see has been gone through under microscope. The first two photographs, these are the microscopic pictures, and the other one is the animation. This is how E. coli looks. Since bacteria comes in multiple shapes, like it can be sphere, and it can be common. Now to the archaea. These are the similar microorganisms as the bacteria, but they are actually pretty different from the bacteria. How are they different? Is their ribosomal RNA? I don't know if you know, but you have to know that the RNA and DNA are actually everything that a being is. And the other property that can differentiate them drastically from bacteria is the living in the harsh conditions. And by harsh condition means the extreme cold and the extreme hot, and that refers to the cold of the Antarctica and the hotness of the volcano. Understand? These are the harsh conditions. Even under actually pretty high pressure, they can survive, which is actually not possible for the bacteria. So the example of the archaea is methanogens. It is actually not the name of the bacteria, it is a group of bacteria that produces the methane gas. Let me tell you the methane gas. Methane gas is a gas which is well actually used in a lot of things. But the one thing that you must be familiar with, or you are familiar with, is the gas that you use in your house to produce fire, in your stoves or in your ovens to cook your food, and that gas maybe came from the bacteria. Wow, who knows? You all know that where methane gas comes from, and it is actually deep in the soil layers, and it is produced by multiple reaction by these kind of bacteria. And since they are so below the soil, so is no oxygen, so it means they can survive in the environment where there is no oxygen, and that environment where there is no oxygen is actually anaerobic environment. Now comes the fungi. The fungi are eukaryote. Here comes a eukaryote organisms. The bacteria and the archaea are the prokaryote, meaning they have no true nucleus, but the fungi actually has true nucleus. And the true nucleus mean it is well protected. Mean just as we say we have a well built house, not just made from some sticks or from nothing, we just call it a home. And the same is the genetic material. Nucleus is home of a genetic material. If it is built strongly, then the organism actually turns to the eukaryote. Now, the eukaryote cells can be unicellular or can be multicellular. Fungi actually has both. The unicellular cells in the fungi are called yeast, while the multicellular are called molds and mushrooms. Actually, just molds, but mushroom is actually very different, so we have to name it separately. Now, how do fungi live? It absorbs nutrients. The example, Saccharomyces cerevisiae. This is a pretty common example, and I think everyone will be familiar with who cooks or know how to cook in their home. And the yeast that we use is actually a living organism, a living, fungi that actually go into our food, and then by food I mean dough made from flour. The carbohydrates in that dough is converted or is a byproduct produce carbon dioxide, which actually fluff our dough and we can make buns from it or bread from it. It is actually a Saccharomyces cerevisiae. It is a yeast and also known as baker's yeast. Means actually very common in bakery, so the name got baker's yeast. So coming to the structure of the fungi, the first one is the single cell yeast. The diagram, and these are yeast actually. These are the single cell organisms. But when you look at the other one, this is actually the fungi known as molds. The mushroom is also the fungi that we can see with the naked eye. Now the virus. Viruses are actually pretty disastrous. They are actually pretty deadly. So one of the viruses is the coronavirus that shook our world with hesitations, or brought the death of the body that it lived in. So it's not just the coronaviruses. Our ancestors that has lived has suffered from the similar viruses that eliminated their whole generations just in the years, few years or few months. These include the dengue virus. The dengue virus, which is present nowadays, is not that severe as compared to the ones that were in past. It actually gave no opportunity for the body to survive, and it just killed them one by one. And these are the diseases that spread human to human as well. So it has to be we have to be very cautious with them. So the example here is the influenza virus, and it is responsible for flu, while the other examples are hepatitis virus, flu virus, and measles pox virus. Now the protozoa, it is a unicellular microorganism. It is also a eukaryotic organism, mean it has the nucleus. True nucleus means the genetic material is protected. Now it can be free living or parasitic. The example is pretty interesting since I think the ones those studied just the biology must have drawn this amoeba or must have seen this amoeba in the 7th, 6th, or 8th class, and it was pretty easy to draw. I think everyone must have drawn it, or maybe not. Well, it has false feet. The extension that is coming out from it. These are called as pseudopodia, and this is just the extension of the amoeba that helps it to move. There are a lot of other characteristics, but this amoeba is not harmful or anything. Now the algae. Algae is a very big help to us. No, not is, but was a very big help to us. We weren't even existing when the algae was existing, and it provided this earth that we are living with. No, it made the hellish environment of the earth to the habitable environment that we are seeing now. Actually, we are contaminating it, but just a few years or maybe 10 years ago, 20 years ago, it was actually pretty healthy. Now, long time ago, there was an algae that was living all alone, and of course it needed food. So, and the environment was hellish. Well, I think not to the algae since it was surviving there, but not to the other organism. Well, actually there weren't any other organism. So it consumed the carbon dioxide and released the oxygen in the environment. It kept producing the oxygen without knowing the importance of the oxygen, neither it needed to be aware of that. So it kept producing it until the atmosphere reached the 21% of the oxygen. And then was the time when the other organisms started surviving. Started growing, and it led to us humans. These are the microorganisms, which weren't even that high quality organisms, but it made the higher organisms to survive in the environment that they were creating or they created. Now there are plants, of course, that produces the oxygen, and there are other microalgae that also produces the oxygen. That or microalgae that produce the oxygen were named as cyanobacteria, and it actually is present in the fresh. So the algae that is given in this example, chlorella, is a dietary supplement. Well, as you can see, it's green, so of course it's producing oxygen. Well, not every green can produce it, but I think in algae cases, the green algae all produces the oxygen. So it is used as a dietary supplement because it has nutrients. Actually, some algae are pretty rich in the protein as well. So if you cannot eat the meat or something like that, you can eat the microalgae, of course, thanks to the microbiology, that we found that there is a microorganism like that existing. So how can we humans leave it alone? Let's move on to the subdisciplines of microbiology. Let's keep it simple and just move it quickly. All the mentioned 5 terminologies are actually the study of the microorganisms that we just went through. Now, Bacteriology, as the name suggests, it is the study of bacteria. Logy actually means to study, and the pre-word that is present before the logy is the one that is being studied. So bacteriology is the study of bacteria, virology is the study of viruses. Mycology, actually they didn't use the word fungus or fungi or something like that. They actually used another language word, which, sorry, I don't remember which language was it, but maybe German, maybe, who knows. There are just few languages that they pick words from. So they use the mycology. So now parasitology. So in parasitology, just the protozoa doesn't come under it. There comes the helminths, and helminths are the worms, the beautiful, beautiful worms that live inside our intestine, or maybe in our blood, or maybe in our tissues, like loa loa, Taenia solium, and yeah, let's keep it simple. So the worms that blocks your intestine, and then your stomach bloats, and then you cannot go to washroom, and then you cannot stool, and then your whole body messes up, and then your hair also messes up. And are the ones that are present in the butthole of the children, or maybe the adults, that keep causing itching, and then we cannot sleep in peace because of these. Then dang helminths, worms, that comes under the parasitology. Now, the phycology is the study of algae. Well, actually, they didn't give the algae the name algeology. They didn't do it. Why? No one knows why didn't they do that. Well, they had to use the words from another language. Of course, English is just so useless, right? In scientific world. So phyco or phyto. So we know that phyto is used for plants. So you just have to remember that phyco is used for the organisms similar to plants, which is the algae. So it is phycology. Now let's study the examples. Like studying the Staphylococcus aureus. Staphylococcus aureus is the microorganism that cause the throat infection. Well, actually, they are present in multiple areas, but I'm uh, giving you an example. And they are actually pretty resistant bacteria to the antibiotics, and it is causing pretty problems, and it is mutating every second day. One bacteriology we research on, like why is it causing bacteria, or why is it causing disease, or how is it causing disease, or how can we cure it, or things like this and that, blah blah blah. And we also study the beneficial side of the bacteria, not just the harmful. Well, virology, just as I mentioned, the this is saying that the research on HIV virus. HIV is a human immunodeficiency virus. By human immunodeficiency virus, it makes our body human immunodeficient. How it makes our body immunodeficient? It's because it attacks our T cells. The cells, well, you don't have to go deep. It attacks our immune cells and destroys them. Well, not all of them, but some of them that are actually pretty important, and they cause our body's immune system to collapse. And if that immune system is collapsed, our body cannot protect itself from other microorganisms that are dangerous. And then slowly but gradually the person gets infected from other infections, and then it dies. Well, you can suppress the HIV virus with medicines or chemotherapies, but you cannot actually kill it. And the person just gets the expends its life 10 years longer or 15 years longer. Maybe it's developed pretty more, but it is actually very persistent bacteria, and it can be sexually transmitted. So it's not just this virus. In virology, we study all different kinds of viruses. Mycology, there is an example of Candida albicans. You can also take the example of mushrooms, or you can take the example of the fungi that grows roots in your soul, or in your feet and your fingers, and then you have to get the operation to remove it. and it is just so persistent that it won't grow. Don't worry though, fungi actually needs an environment that they will grow in, but and that environment is given pretty, and if that environment isn't given, the fungi doesn't grow. So it's your fault that the fungi grows on your feet, and it's because of the moisture and the dirt. And another type is the dandruff. Dandruff is actually a fungi. Not every dandruff is a fungi. It's caused by microorganism fungi, but there is a type of dandruff that is caused by fungi, and that type is a persistent type. That dandruff type is. Now parasitology. It is a study of parasites including protozoa and helms. Well, we discussed it. So let's just go to we discussed the helminth, right? The loa and the intestine one. Well, other ones are, the one that lives in the body and called elephantitis or the things like that. Now let's see the example of the protozoa. That is the Plasmodium falciparum. That's malaria. Malaria is also a disease that wiped quite the population of the world, and it is actually caused by this tiny microorganism that we can't even see with our own eyes. How it works, the Plasmodium falciparum just enters in our body by mosquito, and it develops into an adult. And when it develops into an adult, it causes our red blood cells to be destructed. It causes our red blood cells to be destructed, and then it continues doing that, um, and moving in our own whole body. And then the mosquito comes and sucks blood from us, and then the eggs or the small forms of the Plasmodium falciparum grows into the mosquito gut. Only intermediate life cycle goes into the gut of the mosquito, and then it goes back to bite another human, and that Plasmodium goes and grows in the other human. Now the phycology. We actually studied very in detail the cyanobacteria, algae. So of course, I think you would be pretty familiar with the algae now. Actually, let's study about the harmful side of the algae. There are certain kinds of algae. We call it algal bloom. It causes algal bloom. If the algae grows on the water, and it takes a lot of space on the upper layer of the water and takes nutrients from the water, and it blocks the sunlight, and it takes oxygen from the air and doesn't produce oxygen in return. These are the type of algae that doesn't produce oxygen, and it takes the oxygen and produce carbon dioxide or something. So the marine organisms that are present below them, they just can't get sunlight, they can't get oxygen, and they can get nutrients, and then they die. And this has been done over and over by these kinds of algae. Microbial physiology is the study of metabolic process and functions of microorganisms. Well, we are talking about the metabolic processes. Like, the metabolic processes happening in our body, there are actually a lot of, you can count. Like, for example, digestive system. So of course we have to study the metabolic process of our body, how it does the digestion process and things like that. So the same kind of thing happens in the microorganisms, although they are unicellular, although they are not very complex, but of course they have a little bit of the metabolic processes happening in them. So let's study about the metabolic pathway of lactobacillus species. I mentioned the bacteria that spoil milk and turn it into the yogurt. How does it actually do it? Is the metabolic process of them doing it. Well, you might not understand it completely, but I will just focus on the main points. So the milk has carbohydrates in it. Carbohydrates can be made up of uh, long chains of carbons, and the number can vary, but the ones that are being given here is the hexoses and the pentoses. Hexoses means 6 carbon carbohydrate and pentoses means 5 carbon. So, well, actually, they break down those long carbohydrates and turn it into these, and then they follow the process and produces the following things, and like ethanol, acetate, butanol, or things like that, and produce the yogurt. Actually, when they produces these acetone, ethanol, they actually react with other stuff or combines with other stuff, or they actually are very different things. They are actually very different, and that causes the pH of the milk to change and the proteins present in them to denature, and it actually clumps together and form yogurt. So this is the glycolysis process shown, and they don't go to the Krebs cycle since they are microorganisms. Of course they do the Krebs cycle, but I'm telling you that these species just directly changes the pyruvate and to the acetyl CoA, and from acetyl CoA directly to the ethanol and the acetate. Actually, this happens when there is a lack of oxygen. Well, some microorganisms just follow this path where there is a lack of oxygen or there is not. I'm talking about our case. Of course, because we also digest the carbohydrates, right? Right, we do that, right? Yeah. The microbial genetics. What do you study about the microbial genetics? It is the study of the genetic makeup of the microorganisms and how genes are expressed. In simple terms, everything that is related to DNA and RNA comes under the category of microbial genetics. Well, DNA and RNA of microorganisms all comes under the category of microbial genetics. Well, you will be asking, what do we actually take from studying genetics? What? What? You are actually asking me that? What do you mean? What do you mean by what will you get from studying the microbial genetics? Everything. Everything. You'll get everything from studying the microbial genetics, and here's how. Our cell has a core, and that is the genes, and that is genetic material. By studying the core of by studying the core of your system, you will learn how it works. If you take out that core, nothing remains. Well, of course, it needs the structure and everything, but you must know what the core does actually. So, after the microbes has actually the two types of. The one is the circular genetic material, right? And it is just the DNA that is revolved in a circular position, and its one end is combined with another one, while the other one is just suspended in irregular form or in the form of chromosomes. So the plasmid, the circular DNA, is only and only present in bacteria. You don't have to find it in other organisms. Or maybe I think there is a discovery that they have found the circular DNA in another microbe, but it actually is just the major property of the bacteria. Anyway, the chromosomes are of course present in every kind of organism, whether it be prokaryotes and eukaryotes. So we were talking about the circular type of DNA, right? And that is the plasmids. We call them plasmids. And if we study these plasmids, it actually is the cause of the bacteria that they become resistant, or they develop uncertain habits that we don't see in the bacteria. when we studied before, and then we study after effect. It the plasma due to the plasmid release and the stressful conditions, it causes the change, and this type of genetic material can be transferred from the one bacteria to another. Well, not all, but there is a way of transferring it. Unlike the chromosomal genetic material or chromosomal forms of the genetic material, it cannot be transferred. A simple example of the property of plasmid, it is the Bacillus thuringiensis is a bacteria, and it has a plasmid, and its plasmid has ability to produce toxins, okay? But don't worry, the toxins are not for us human beings. It is for the organisms that causes distress to the human beings. So of course we have to get rid of them. Well, it's actually pretty cruel, but I think they cause really problems. So of course we have to go to use the harsh. Well, so the toxins, the diagram, the first diagram is actually the plasmid of the Bacillus thuringiensis. There is actually not given the right coding. It's just the main points where we can cut the plasmids or things like that. So you don't have to study it in detail. It's not all the plasmid, and you won't find the genetic codes on it. The next figure is the insects. Well, it causes toxins, of course, but it causes for the organisms, and those organisms are insects and bugs, right? So turn towards microbial ecology. Now, the study of interactions between microorganisms and their environment. I think ecology is a topic that you all should be familiar with, or no? I think you are all familiar with, since you have been studying it from childhood, of course. But I'm sure you must not remember. I'm sure you don't remember anything particular. Well, I'm not saying that you didn't study it, but well, anyway, the study of how nitrifying bacteria. Well, of course, we are microbiologists. We are not just studying about ecology. Ecology is actually the study of ecosystem. And what is ecosystem? Ecosystem is the environment where the living things and the non-living things interact with one another. So this is the ecology where we see the living organisms are the microorganisms, and how these microorganisms actually interact with the higher organisms and their environment, and how do they play the positive role and play the negative role. So the following example is just about a bacteria, right? Nothing important, right? That's why we are studying microbiology. Anyway, so it's a nitrifying bacteria, or for example, the nitrosomonas bacteria. Well, to talk about the nitrifying bacteria, we have to know about the nitrogen cycle. The diagram shows that the nitrogen exists in two or three forms. That is in the atmospheric nitrogen, means nitrogen gaseous form. Then it is in the form of ammonia, like NH3. Then it goes into the nitrates and nitrites, which is a usable form of the nitrogen. We or plants or other organisms, well, unlike bacteria, can't use the nitrogen directly from the atmosphere. So we need someone that will change that atmospheric nitrogen into something we can use, or something the other organisms can use, and then they convert it for us to be usable. So we need the microorganisms that will turn our nitrogen into the ammonia first. Of course, no one else other than the microorganisms can use the unusable form of nitrogen, that is the gaseous. So they change it into ammonia. But ammonia is still not being able to be used by the organ. Now the ammonia is converted into the nitrates and then the nitrites, and that is actually done by the nitrifying bacteria. Nitrifying bacteria. Now, the nitrifying bacteria gave us actually a present, and now that is used by plants and fungi and other bacteria, and then the plants are used by or eaten by the herbivores, and the herbivores were eaten by the carnivores. What is India? And the carnivores were eaten by the omnivores. So omnivores has us humans, and we humans actually get to eat that nitrogen or use that nitrogen. Actually, I didn't have to go that long road. We can take it directly from the plants. So that was just the role of the microbe in the environment, ecology, and specifically nitrogen fixation. They has role in the carbon cycle, oxygen cycle. They have a lot of roles. Now let's go to the pathogenic microbiology. Well, everything related to disease causing microorganisms comes under the pathogenic microbiology. Example is, well, we have studied plenty of examples, but let's go to another one. HIV virus. I'm not talking about the bacteria, okay? In pathogenic microbiology comes fungi, viruses, plasmodium, no, I mean parasites, and bacteria, archaea, everything comes under this. So Mycobacterium tuberculosis. This is the bacterium that causes the tuberculosis. What is tuberculosis? It is a disease of lungs. The bacteria goes to the airways, and in the air they goes to the lungs. From the air pathways they goes into the lungs, and in the lungs we have immune cells known as macrophages. They enter the macrophage and kills them, and slowly they colonize our lungs, and then they dominate our lungs, and then they destroy it. And when they destroy it and our lungs don't function, and then we die. Actually, that is the severest case that can happen, but actually it damages our lungs. So this was just an example of pathogenic microbiology. Now the immunology. I think people find immunology complex, but I find it interesting since it feels like a game of enemies coming to fight, and then our hope causing cells, immune cells, goes and fight with them and protect us. You know, it's actually an action game. So in immunology, we study about how to respond, how the immune body responds to the immune function. Like it's like how we respond to our enemies, right? But not on the macro level, but on the micro level. So there is one research example is vaccines against viruses like measles or influenza. Below is the virus pathways that just shows how we fight against it, right? So it doesn't happen da 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 and everything happens. It is actually a pretty complex process that this I don't think this diagram has shown it is that complex. The viral antigen enters our macrophages, right? The macrophages we talked about, and they enters into it, right? And then our macrophages will do what? They make an identity card of the viruses, and they put it on the surface of their cells. Then the other cells, immune cells, our action cells comes, and specifically the T cells. Remember the T cells that HIV virus infects and destroys. Those type cells actually comes and kills those macrophages and all the cells that has that identity card of that virus. So actually the T cell recognizes that these cells have viruses, and they will actually create more viruses, so we have to get rid of them, right? That's why the identity causing immune cell, or the identity creating immune cells, those are aka macrophages, is very important in the immune response. And of course the T cells that comes afterwards and kills the now the T cells not just kills the cells that has viruses in it, right? Of course, there are still viruses that are outside the cells. How will we deal with that? And there comes another type of cells, and that will be B cells, right? Now, B cells are the helper of T cells killing the viruses. B cells actually makes small bodies, so every small body is specific towards the specific virus. And B cells have another function, not just directly killing the virus, but actually causing memory cells, the cells that has the memory of the virus. So if another time the same virus comes and attack them, they will be standing and protecting, and they will say goodbye, don't come, you don't belong here. So this is how actually we create vaccine. We have the unkilled or maybe killed or maybe living organism viruses in these vaccines. Well, and we inject this in our body. It actually prepares our body to fight viruses. So if a stronger virus of that type in our body, we can protect ourselves. So we don't study just vaccines, we also study antibiotics and different kinds of things in the immunology, since well all the microorganisms causes the action of the immune system or causes the activation of the immune system. So everything related to immune system is studied under the immunology, and not just the relation or not just the two sides facing one another like microbes and immune system. We study the immune system separately to understand how actually immune system works. Now come towards the industrial microbiology. This is actually the practical side of the microbiology. But the thing is, I don't find it very interesting. Well, these are just machines, and these are just not my type. Just industry. Anyway, I'm just kidding. Everything microbiology. I'm a microbiologist studying microbiology, of course. Whether I have to. I have to go to like everything to actually keep these in mind. Now, the industrial microbiology. Industry is about everything: fabricating clothes, creating clothes, shoes, food. So why not microorganisms? Why not create an industry of microorganisms or microorganisms related industry? So what we do actually is we use these microorganisms in the industry on large scale. We multiply them in a large amount, and then the and these microorganisms actually work as labors to produce what we need. Take simple industry as an example. We have to create food, right? What do we need? We need actual workers, humans, to do that work, or maybe machines nowadays. But no, for other for to operate machines, of course, we need still humans. So we take humans and they make things we need in the industry. So in industrial microbiology, we take microorganisms and they create stuff for us on larger scales. There's an example and that is the use of the fungi Aspergillus niger, and that creates the citric acid. Well, you will be saying, what is citric acid? Why do we have to create it on large scale? Why do we need it? Citric acid. Do you know citric acid is actually in the vinegar you use in your home? The one that is actually vitamin C, or the citric acid that is present in a lemon. You actually use it. You actually need it. There's always one in your home, and you know it. So where does that come from? And it comes from the industries. And which industries? The industrial microbiology industries, the one where microorganisms are used to produce citric acid. The diagram shows the Aspergillus niger, and the right side shows the industry where the citric acid is produced. The middle part shows how we give sugar to that fungi, and it produces fructose 6 phosphate and then fructose 1,6 diphosphate, and then it produces pyruvic acid. This is actually the glycolysis process, and then it goes to the acetyl CoA and then goes to the Krebs cycle, right? And when it goes to the Krebs cycle, it produces the citric acid. We all know that the Krebs cycle has a component that is the citric acid. Anyway, this is just an example. We can produce algae, the one that we can use as a dietary supplement in industries as well on higher scale. Actually, it is already produced. We don't need to make it. I mean, you can create your own industry and use the microorganisms. Actually, there is an interesting thing about industrial microbiology, and that is, the microorganisms. Actually, it is in combination with the genetic engineering, or you can say genetics, since these uses the microorganisms to degrade the plastics that are contaminating our whole world and cannot be digested, degenerated, or degraded in any case possible. So we use the microorganisms for that. Actually, there are no microorganisms that can degrade plastics on a normal basis unless we design them in the lab and then give them to the industrialists and then tell them to take all the plastics and degrade it. Actually, I think it will become very common in the coming future, since well, plastics needs to be eliminated, otherwise we will be eliminated, okay? Plastic needs to be eliminated or we are going to be eliminated. Now comes the environmental microbiology. Environmental microbiology. Again comes the environmental. What is the microbial ecology and environmental microbiology? Actually, it has to do with one another. These are two different things. Why? It's because, let me explain. In microbial ecology, we just see how the microorganisms give to the environment or take from the environment. We don't actually study how inside of those microorganisms work, right? How actually their processes work to give to the natural environment and things like that. Like inside their gut, how do they actually take that nitrogen and how actually do they um, process that and convert it into ammonia or something like that. But in environmental microbiology, we actually dive into that. Actually, those two are not that different kinds of things. So here, see, there is symbiotic relationship. Symbiotic relationship is actually a relationship where both parties are benefited. It's not like parasitic relationship where one body is harmed and another is benefited. Okay, there are microorganisms like trichonympha and flagellates, and they live in the gut of the wood-eating mites. They actually produce cellulase that digest the cellulose in the wood, and the mites that eat the wood actually don't have the mechanism to digest that wood, but and they would die if they didn't have that microbes, right, living in their symbiotic relationship. But thanks to the microorganism, they can digest that wood by cellulase enzyme. And you'll ask, what will they get in the return? They get nutrients in the return, which they couldn't get when they were living alone, or maybe they cannot live in the environment present there, so that's why they chose to live in the gut. Here comes the food microbiology, and it is the study of microorganisms and the food, including spoilage and the fermentation process. We all can't live without food, and microorganisms need food as well. So how do their food resources match with our resources, and how do they connect, and how do they cause multiple phenomena? And those we are studying in the food microbiology. The Saccharomyces cerevisiae produce carbon dioxide in the food dough and causes the dough to puff up, and then we put it in the oven and then it gets into the bun, thing like that. this food spoilage, like we study in the fungi mycology, right? That the fungi mold actually grows on the bread and causes spillage. Or actually, fungi also grows on the potatoes that are present in the moist environment, or the bacteria that cause the spoilage of the tomatoes. Clinical microbiology, the study of microorganisms in a clinical setting, focusing on the diagnosis and treatment of the infections. For example, identifying the streptococcus pneumonia in the patient samples to diagnose pneumonia. Actually, we have to study the steps that actually happens in the clinical settings. So there are actually three main levels that we can go through to identify the microorganisms. Now, the first one is the microscopy. Of course, before doing everything, we have to go to the microscopy method, right? We just have to take the sample of the microorganism, and then we have to go to the staining, like coloring it, since we can't see invisible stuff, right? Or maybe it's fungi, so we don't have to color it, but we have for a definite look. So anyway, we color it, or in scientific term we stain it, and then put it on a slide and under the microscope, and we study its structure, physical structure, and we can diagnose it from that. Actually, before microscopy, we have to grow them, culture them, and then we have to study its physical appearance. Like in case of bacteria, if it grows on the plate, agar plate, the one that we have grown on, it creates colonies. The space where the bacterial cells are congested is actually the colonies, and it becomes colonies only when the same type of bacteria are close together. So we can study the bacteria from colonies. Some bacteria creates larger colonies, some creates colonies size of dot, or some bacteria has colonies that are concave, some has convex, and some are puffy, and some are flat, and some are colorful, like yellow, whitish, or greenish, brownish. Like we can actually categorize them based on their colors. Like we can shorten the spectrum of our diagnosis for further diagnosis. And after that, we stain them. And then under the microscope, we study more of their characteristics on cellular level, like their cell wall, or they have flagellates, or or do they have, what kind of organelles they have, or what kind of nucleus they have, or things like that. After these things are done, and we still are not sure what kind of microorganism this is. I mean, we have shortened the spectrum, like its shape is rod shape, so it must be bacilli. But what kind of bacilli actually? So even if we go into the bacilli bacteria, we still will get to a point where we won't know that the bacteria is what. So after that, we go after the chemical methods, and in that we do the catalase test, oxidase test, and triple line test, and tests of, like, there are multiple. There is actually a list of that, and we will cover that in another video sometime. Now, the antimicrobial agents, like we studied in the immunology, we don't just focus on the vaccine or antibiotics, but there's actually a different field in which we actually focus on each and everything of the antimicrobial agent. Where did it come from? How is it created? What will it cause? And things like that. So this that increase the growth of microorganisms, including antibiotics and disinfectants. Now, there is a difference between antibiotics and disinfectants. Both the antibiotics and disinfectants kills the or stops the growth of the microorganisms. But the only difference is antibiotics are used for living organisms, on different disinfectants for non-living things or the surface of the living organisms. The example is of the antibiotic, common antibiotic is, and the first antibiotic that came into its existence and discovered is the penicillin. It is actually the antibiotic that comes from the Penicillium chrysogenum. And this was discovered by who? Alexander Fleming. Below is the process of industrial production of this antibiotic from Penicillium chrysogenum. And actually, there are different penicillium that can produce this penicillin. And there's actually now the multiple generations or multiple types of penicillin. And there are cephalosporin and tetracycline. Now the biotechnology. The application of microorganisms in biotechnology such as genetics, engineering, and bioremediation. Or the application of microorganisms in technology. There is a difference between application of microorganisms to produce antibiotics or to produce, the citric acid or things like that in industrial biology. This is different. This is the microorganisms used in technology or technologies used on the microorganisms, such as genetic engineering. This is pretty concise, and genetic engineering is a common theme of the biotechnology. In genetic engineering, we do what? We usually take the plasmid. Since plasmid is small in size, so it won't be overloaded, and we cut it and take the genetic material of some other organism which has the property that we desire to be, and that microorganisms. We combine their genetic material and another characteristics appear, in scientific words, another phenotype appear. Of course, the genotype is changed by us humans, and the phenotype appears by the microorganism. And that was the phenotype that was desired by us. For example, the bacteria didn't had the penicillin producing ability, but we needed that. So we just took the genetic code from that could produce the penicillin and took it into the placed into the other microorganisms. Now you will be asking, why do we have to take it from the other organisms? Why can't we use the other microorganism as it is? There are multiple reasons. Machinery is too difficult, or it needs a lot of nutrients, or its life cycle is very long, and things like that. So we select the microorganism that is very low cost, like it don't need nutrients that much, or it can live in harsh environments, or it doesn't need that much pH maintenance or things like that. And then we just have to put the genetic code. The microbial taxonomy. This is pretty ugly, actually. This is the actual scientific field or scientific discipline, and it makes me very sad, you know. Anyway, let's study the microbial taxonomy. This is everything that we humans study. We name them first, right? We name them to distinguish it from another thing. Like we have to tell something that we're talking about. We have to name that thing. That's why the scientists put so much effort making the names of the things unique from one another. You cannot find an organism that has a similar microorganism in scientific world. They all have different names. That's the first step in microbial taxonomy. And the other, the microbial taxonomy name the microbes in two terminologies like the genus and the species, just as your family name and your own name. It is more clarifiable and more distinct and give us more chances for it to be west. So after we name things, we have to categorize the things. Like we cannot, if we have to talk about a lot of things in our home and we are telling this to someone, we cannot just name everything. That will overload the person and the person won't know what we are talking about. That's why we have to group them. Like we have to group the similar thing in one group, and then instead of talking about all of the things one by one, we just have to name that one group and we'll know what we are talking about, including everything. So that's why the organisms that have similar characteristics, we put them in the same group. And it starts from higher levels to lower levels depending on the characteristics, similarities, differences. The highest category or the most general category is deemed as a life category, all the living organisms. After that comes the domain, kingdom, phylum, classes, and one by one everything until we reach a point where the organisms is actually very distinct and there is no way to classify the organisms any further. It only occurs and which we call the subspecies if there is a different, slightly bit difference in one to another. Like it just has one different characteristics. All of the other characteristics are same, just one characteristic is different. That comes under the subspecies category. So anyway, with microbial taxonomy, we all deal with classifying and organizing all of these things to use the microscopy techniques. You must be telling that when we study the microscopy techniques, right? Why? Why do we have to study? It's simple because it's microorganisms. We need microscope, so of course we have to microscopy techniques study, and that's why we put it in the separate class. So the methods used to visualize microorganisms include light microscope. You know that there are different types of microscopes, and every microscope has the capacity to study visualize the structure of virus. For example, using electron microscope to visualize the structure of rabies viruses. Since the viruses shapes or the microbes shapes varies, no, not shape, size varies. So of course it will need the more and more complex microscope for the smaller and smaller microorganisms. The culture techniques, the clinical we were studying just earlier, and we covered the microscopy, and then we are coming towards the culture techniques. Culture techniques difference means that we are growing bacteria in a lab setting, but not just bacteria, microorganisms, of course, all everything. So for example, growing them in a liquid media is actually the material that has nutrients and is used to grow the microorganism, and it actually has everything packed in it which the microorganisms would need. So in culturing techniques, there are a lot of techniques like plate method, plate spreading method, enrichment culture method, and streak plate method, and things like that. Or maybe culturing on the solid media and culturing on liquid media, or culturing in suspension media, or serial dilution methods. If you wanna study them more, you can comment below to tell me. We used all the images from the Google, so I didn't took these images or created these images. So if you wanna find them, you can all go to the Google and just search for the example. that I have given the images below it, and you'll find them. So here I take my cue to leave, and that is it. Bye bye.